I would like to welcome Jose Ceballos. Jose is the division chief of head and neck surgery at Washington University in St. Louis. He's also a physician scientist and runs an NIH-funded lab. He's going to be talking about his research regarding smoking and HPV and epidemiology in cancer biology. With no further delay, Jose, the stage is yours. It's a pleasure to see you here.
Thank you, Tulio, very much for the invitation, and thank you all for having me. It's really a tremendous honor. I have a lot of friends in the audience. Tulio and I go way back to, he was my fellow when I was a resident, trying to get through Texas Children's. Chris Holsinger has been a friend and mentor for a long time from MD Anderson. I know Eben, I know John, and Paul Zolkind, your current fellow, is one of our star graduating residents who actually contributed to some of the work I'll talk about today. So it's really a huge honor to be here and to talk about some of the work I've been doing really over the last 4 or 5 years in understanding smoking and HPV positive oropharynx cancer. Really looking at this holistically from the point of epidemiology all the way to understanding how smoking impacts HPV cancer, tumor biology, and outcomes. I have a couple of disclosures. I'm a founder of a small molecular startup company called Droplet Biosciences and an equity shareholder for another very early stage company called Summit Biolabs. And I won't be discussing anything in this presentation. So today, what I wanted to talk about is how smoking impacts the entire arc of HPV positive oropharynx cancer, from its effect as a pro-inflammatory and immunosuppressive on infection, oral HPV infection, all the way through cancer treatment and outcomes. And I think this touches on epidemiology, the way we treat this disease currently, our understanding of genomic heterogeneity in the cancer, and the tumor immunology. And then I have some conclusions and next steps. I think it's important to look at this holistically because, oftentimes we get into a trap of thinking that HPV positive oropharynx cancer is a disease of young non-smokers. And I think that was kind of the prevailing theory early on, but we now know, as I'm sure you see in your population, and we certainly see in ours, that we see HPV positive cancer across the entire spectrum with respect to age. We also see HPV positive cancer commonly in smokers, in current or in former smokers. We also do see our share of never smokers, but it's an uncommon population, at least here in St. Louis. And I think there's a lot that we're learning. Although we know this disease has excellent oncologic outcomes, generally speaking, we have to be cautious as we implement new de-intensification trials because there are certainly patients that don't deserve de-intensified therapy. And I think this has come to light in some of the published initial de-intensification trials that are coming to completion now, where we're identifying certain populations where this approach is not appropriate. We do know that it's increasing in incidence, and I think this slide has probably been shared more than many in talks like this, where the percent of HPV positive cancers has increased by over 200%, and we've seen a significant decline in HPV negative oropharynx cancer. And as we all know, that the smoking rates in the US have decreased significantly during the same time period, but not to say that the HPV positive cancers aren't being at least driven, or that there's some interaction between tobacco use and HPV virus, both at the point of carcinogenesis all the way through response to different therapies. So we know that smoking increases the risk of oncogenic HPV infection, and I think that's been well characterized, as I'll mention in my next slide. What's less clear is how this happens. Smoking, as we all know, has over 60 different carcinogens. It has a wide impact on everything from DNA repair to apoptosis to oxidative stress. And understanding how this relates to carcinogenesis is somewhat clear in HPV negative cancer and not very clear in HPV positive cancer, as I think I'll show in the next couple of slides. We do know that tobacco impacts innate immunity and adaptive immunity in different ways, and depending on which paper you read, you find that there's an increase, for example, in cytotoxic T cells in smokers in the local oral immune environment. But we also see a decline in T cell infiltrates in smokers with HPV positive cancer. We also see differences in pathways in terms of interferon gamma NK cell function, and it really there's mixed data on whether this is a pro-inflammatory and immunosuppressive, or and how it actually impacts infection rates. We all are familiar with the way HPV goes from acute to a chronic infection to cancer. There's a long latency period, and there's a productive period where the virus actually infects the crypts of the tonsils and the base of tongue, followed by viral shedding. There's a persistent infection period that leads to cancer over the course of 20 to 30 years. And where along this pathway smoking has its impact remains relatively unclear. There are data showing that smoking will shorten the persistence or the latency period from virus infection to cancer. We know there are data that smoking causes an increase in oral HPV infection, oncogenic HPV infection. And we know there's some temporal relationship between when people start smoking and when people are exposed to HPV for the first time. These are happening around the same time, but we don't have the luxury of going back in time and understanding where these interactions take place and how smoking leads from acute to chronic infection and persistence. But these are open questions that I think have a bearing on our understanding of carcinogenesis and treatment response down the road when these become cancers. So Amber D'Souza and Carol Fakhri published a paper, which was a secondary analysis of NHANES data. NHANES is a large population cohort of a representative sample of the US, where they looked at oral HPV infections in a very large population of folks and looked at different characteristics that define risk and risk profiles. And they published this very nice diagram that tries to personalize the risk or provide a personal risk of oropharynx cancer. As you can see here, there's an interaction between number of sexual partners, risk of HPV infection, and smoking. So the highest risk people were men who had greater than 5 sexual partners and a greater than 10 pack year history of smoking. And that risk was about 14.5% or 14.9% risk of having an oral oncogenic HPV infection. Now, we don't know, as we all know, it takes 1 sexual partner. It doesn't have to be 5, but 5 probably represents a higher risk of exposure. Why 10 pack years versus 20 pack years versus 30 pack years is an unanswered question, but we know that there's an interaction there causing an increase in infection. If we fast forward 20 to 30 years, we also know that smoking impacts outcomes for people who develop chronic infection into oropharynx cancer. And this was work from Dr. Ang at MD Anderson showing for the first time that there are 3 different risk groups of oropharynx cancer: low risk, intermediate, and high. The low risk being low volume tumors with a less than 10 pack year history. Intermediate risk comprised primarily of HPV positive patients who also smoke. And then the high risk patient population were HPV negative tumors primarily. And you can see there was, they demonstrated for the first time that there was a significant difference in outcome based on smoking status, overall survival in this case. These data have since been replicated multiple times in multiple cohorts, retrospectively as well as prospectively. These are data from Maura Gillison, where they looked at smoking, the effect of smoking on outcomes in RTOG 9003, and again, RTOG 0129. And they show that there is a significant difference in overall survival when you look at 10 pack years, hazard ratio of 2.1 to 2.5. The paper on the bottom here is a recent paper by Vlad Sandulak in Houston, where they looked at a very large cohort of patients at MD Anderson and found that 30 pack years was the cutoff. And frankly, I'm a little suspicious whether those 30 pack year patients are truly HPV positive. But all this to say that head and neck cancer or HPV positive head and neck cancer has been impacted in terms of survival by HPV. But understanding a variable like this, which is a patient-reported variable, is hard to sometimes quantify. And I think part of the thesis of what I'm talking about in this talk is that we could probably do better than simply counting pack years or having folks recall what their pack year history is when we're making big, important decisions about how we're going to treat this cancer. So we looked at this in a very large population-based cohort in North Carolina when I was at UNC, the Chance Cohort, the PI of whom was Andy Olshan. We found that among patients who survived greater than 5 years after their cancer diagnosis, and they were smokers, they continued to have worse outcome than cancer-free controls. And I think this is driven by competing causes of mortality related to smoking, or treatment effects, but we do see that patients who had cancer and survived 1 to 5 years had a worse survival than folks that did not have cancer. So our understanding of how smoking impacts HPV positive outcome has influenced significantly the clinical trial portfolio or profiles that we have across the country. And largely, treatment de-intensification has focused on non-smokers or very low smoke pack year smokers. This phase 2 trial from Mayo Clinic, HN002, and now HN005. All of these trials that are important trials that are now demonstrating results are focused on a group of patients that are the lowest risk patients among HPV positive cancer and don't necessarily represent the majority of our patients. So we have to wonder about generalizability of these results as we move forward in the era of treatment de-intensification. But more importantly, I think we could probably do better than understanding just tobacco. We should understand why and how tobacco is impacting the biology of these cancers and how that could potentially influence our treatment strategies. So the first question is, what do we know about the genomics of head and neck cancer? And in the last 10 years, I think we've learned a tremendous amount about driver mutations and genetic alterations that lead to HPV negative and HPV positive head and neck cancer. The TCGA that was published, Neil Hayes is one of my close colleagues and mentors, found that P53 mutations were the predominant mutation, driver mutations in HPV negative cancer. They also found canonical mutations that we now consider canonical to be HPV negative mutations, STAT1, NOTCH1, PIK3CA, HRAS mutations, AJUBA mutations. However, it's important to note that in TCGA, among the 479 tumors that comprise TCGA, the first cohort that was published included only 39 HPV positive tumors. And this probably relates to the fact that HPV positive tumors were typically biopsy specimens and not surgical resections. And I think doing those studies even 5 years ago was very different than what we can do from paraffin now in small samples. But TCGA didn't shed very much light on what we know about HPV positive cancer mutational status. Several papers have been published since, including this one by Maura Gillison from Genomics Medicine, giving us a better understanding of the landscape of mutations in HPV positive cancer generally. We know that 30% or so of tumors have PIK3CA mutations. TRAP3 is a common alteration, zinc finger 750, MLL2, FGFR3. These are common mutations that we now understand to be drivers in HPV positive cancer. But even then, with the numbers that we have, we don't quite understand how smoking impacts these outcomes. So our data are limited in understanding this intermediate risk group, which in my population comprises a majority of our patients. So there is an understanding of how tobacco leads to mutagenesis in cancer globally. And this comes from our understanding of these two trinucleotide signatures. These are patterns of mutations that are primarily base substitution indels that form these mutagenesis patterns that we can look at tumors and understand that the DNA repair pattern we see among these particular cancers are driven by smoking, for example, or in the case of HPV positive oropharynx or cervical cancer, Epidemic associated signatures are driving the majority of these tumors. Any given tumor can have different proportions of signatures. For example, as you can see in the top slides, you could look at lung adenocarcinoma and find that the majority of the mutations in any given tumor are driven by the tobacco signature 4, but there are some of their mutations in any given tumor driven by the Epidemic signal or the age-associated mutational signature. And so we understand, we can look into these tumors and understand what's driving mutagenesis. And one could theorize that an HPV positive cancer in a smoker may have more of these signatures associated with tobacco use, like a signature 4. But we don't know largely yet whether or not this is true, whether the HPV positive smokers have different patterns than HPV positive never smokers, nor do we know whether HPV positive smokers develop mutations associated more with HPV negative. Do we see P53 mutations among the HPV positive smokers or KRAS mutations that we would expect to see more in HPV negative. And with that background, that's kind of what we set out to do when we looked at this in a large cohort that we've built over the last 3 or 4 years and that we're preparing a manuscript for now, hopefully to be published this year. We looked at the largest cohort of head and neck cancers assembled. We combined 4 different cohorts from 4 different institutions, including UNC, WashU, the Chance population-based cohort, and LSU Shreveport, to include 531 tumors. Importantly for this talk, 259 of these tumors were HPV positive, and this is by far the largest cohort of unique HPV positive cases has been previously defined. We used a targeted sequencing approach and included 770 genes, cancer-associated genes for this cohort. And importantly, we were also able to sequence the virus with significant depth, 100x depth of coverage for the entire HPV genome. And this allowed us to understand structurally what HPV looks like in these cancers. And our goal was to identify differences mutationally between HPV positive cases in smokers and non-smokers. And there were some secondary aims to define whether viral structure or viral copy had prognostic significance, and whether we can define better biomarkers for how we look at treatment decisions in HPV positive cancer. So I'll go through some of our data, and this, as I mentioned, includes about 259 HPV positive tumors. And we looked at the mutational profile across different variables. We looked first and foremost across smoking, PAC years, current, never, former. And to make a long story short, we found that there were no significant differences mutationally between smokers and non-smokers. And we looked at this every which way we could. We looked at this a priori. We looked with non-supervised clustering. We looked at this using different categorical variables of smoking, current, former, never, PAC years, 10 PAC years. And we really couldn't see a signal that showed genomic differences between HPV positive smokers and non-smokers. But what we did see is we started noticing that there were potentially patterns related more to the HPV virus, to the structure and function of the virus, that could help us understand genomically the differences in these tumors. But importantly, no mutational differences. PIK3CA, MLL2, and MLL3 were the more common mutations in these tumors, irrespective of whether they were in non-smokers or in heavy smokers. We also looked at copy number differences, and this is a nice plot looking at copy numbers across the genome between HPV positive and negative. The black line represents the difference, the red line represents positive, and the blue line negative. And so wherever this black line deviates from the baseline 0, there is a copy number difference between positive and negative. And some of the ones that stood out most readily included chromosome 1, where Notch2 and NRAS are present, and then the Q arm of chromosome 3, as well as chromosome 7. So we know that we can detect differences in copy number between HPV positive and negative. We then asked the same question between HPV positive smokers and HPV positive non-smokers. And again, this is a large cohort of almost 260 tumors. And we really didn't find any significant differences in copy number variation between positive smokers and positive non-smokers. Again, we did see these mutational differences between positive and negative, HPV positive, HPV negative, but no real genomic differences by copy number in smokers and non-smokers. We then went back and asked about mutational signatures. And as I mentioned, these trinucleotide signatures give us an understanding of the mutagenesis. And we thought, well, are we going to see more base substitutions associated with tobacco among the patients who are smokers? And the short answer is we did not. In fact, we did not see a single case, even in a cohort that was skewed towards heavy smokers. We did not see a single case where the tobacco signature was present in this entire cohort. So age, the APOVEX signature, and the double-strand break repair signature were the predominant ones, and not a single patient had a tobacco-associated signature. And I'll mention that this tumor cohort included patients with up to 30 years PAC year history with smoking and confirmed HPV-positive cancers. What we did start noticing, though, was that there were differences with respect to mutagenesis or trinucleotide signature when we looked at different levels of HPV copy in the tumors. So low HPV copy or viral copy really sequenced HPV reads that we can get in our sequencing. had different profiles where APOBEC signature declined as we got into higher HPV copies. So this was a clue that the virus may be driving mutagenesis, or the mutagenesis is driving the virus. And then we wanted to take it one step further and ask about HPV structure. So again, as I mentioned, we had the luxury of 100x sequencing of the entire HPV genome. And we're all very familiar with episomal and integrated HPV subtypes, where integrated subtypes make up about half of HPV positive cancers in oropharynx cancer, and more, about 80% in cervical cancer. And we were able to see both those subtypes, episomal on the left, integrated on the far right. And what you're looking at here is a circus plot where we have human chromosome 1 through Y along the outside of the circus plot. And then here is the HPV genome. And you can see different HPV genes, E1 through E7, as well as the latency genes. And this line represents an episome, a circular structure that links this HPV genome. So this was an episomal case. And we saw this in approximately 50% of our tumors. Integrated cases, this particular case has evidence of integration with an integration point in chromosome 8. We saw multiple integrations. We saw different variations, as you can see here, of tandem repeats of partial HPV in the integrated cases. And then we saw this unique case. These were rearranged cases that comprise about 15 to 20% of our cohort, where not only are we able to see episomes, but we see that there are these tandem repeats of parts of the genome. And Maura Gillison described this in cell lines, or something similar to this, where they had a looping structure of tandem repeats of the HPV genome. But this is unique because these are episomes, and they're not patterns of integration. So we call this a rearranged subtype, and we started exploring the significance of this, not just in this cohort, but in others. We also found significant copy number differences by structural subtype, where rearranged, integrated, and episomal types of HPV had differences in important copy number regions of the genome, including in 3q, as you can see here, and 11. Now, the reason I bring up the viral structure in part is because we didn't find what we were looking for. We didn't find tobacco differences that we were hypothesizing, but this did lead to an interesting novel finding of the importance of viral structure and viral copy as we think about prognosis in this cancer. So this is the same cohort, and we looked both at pack year history of smoking, and we looked at HPV structure and viral copy. And we found that looking at the viral structure in combination with viral copy was significantly more prognostic than 10 pack year history, as you can see here on the left. And specifically, the high viral copy cases, the high viral copy integrated cases had significantly better outcomes than the low viral copy integrated cases. And we've now replicated this in another cohort here at WashU, also part of this, this pending manuscript that we're writing. This requires a lot more exploration, but I think at the very least, it tells us that beyond understanding something like smoking, which is a difficult variable to classify, we can find biologic subtypes of this cancer related more to the virus that probably lead to differences in outcomes. So as I mentioned here, this low HPV copy integrated subtype appears to have significantly worse outcomes. So getting back to the smoking question. So the fact that we found no differences in driver mutations or copy number variants, even in a cohort that was skewed towards heavy smokers, leads to the next question. Well, what is it about these tumors that's different? What else could be happening? And the natural answer, of course, is that there are differences in tumor immune response. So there is evidence from other cancers that show this, including in HPV negative head and neck cancer and lung cancer. We know that there's this interesting, this is a paper from Luke Morris at Memorial, where they looked at TCGA data, DNA sequencing data from TCGA, as well as in some other new cohorts at Memorial, where they noticed that there were differences in the low smoking signature versus high smoking signature. So going back to the smoking trinucleotide signatures, patients that had an HPV negative cancer who had less of a proportion of smoking signature had significantly better outcomes than high smoking signatures. In lung cancer, there were really no differences by smoking signature with respect to outcome. But where it gets really interesting is that there's this paradoxical relationship between smoking tumor immune infiltration, where patients who are smokers in head and neck cancer tended to have a lower immune infiltrate based on their RNA-seq data and these immune scores that they developed, or IF and gamma pathways. So smokers with HPV, with head and neck cancer, had less tumor infiltrate based on their RNA signature, but smokers with lung cancer had significantly higher tumor immune infiltrate. And smokers with lung cancer tend to respond better to immune checkpoint inhibition than smokers with head and neck cancer. So this was an interesting finding because it tells us, again, going back to my first slide about the tumor immune microenvironment and how it affects infection, smoking has a variable impact based on tropism, based on where this cancer is, based on what cells it's affecting, and based on the local immune microenvironment that we have a lot more to understand on. Importantly, this analysis, again, didn't include a lot of HPV positive cancers. So we're still left asking, what's the effect on HPV positive tumors? How does smoking and the immune infiltrate work in this cancer when we see such differences between lung and head and neck cancer? So others have looked at this in oral cavity cancer. There was a paper actually from probably 15 years ago now that Jeff Myers and Curtis Pickering published, showing that in oral cavity cancer among non-smokers, there were no significant differences in terms of genomic variants or driver mutations in oral cavity patients among smokers and non-smokers. And this group found a similar thing. This is a much more recent paper from France showing that the genomic profile of smokers and non-smokers with HPV negative oral cavity cancer were not different. What was different in these 4 different data sets that they looked at was PD-L1 expression, for example, where PD-L1 expression pembro response signatures were significantly enriched in the non-smokers compared to the smokers. So this gives us a clue that smoking has an impact on PD-1 expression and the potential for immune checkpoint inhibition. What about HPV positive cancer? This is another paper from Vlad Sandulake in Houston, showing that there were differences. If you look at simple IHC, this was done in a big cohort at the Veterans Hospital in Houston. They found that CD8 infiltration was associated with improved outcomes and was also associated with tobacco exposure. So patients who were HPV positive and smokers had significantly less CD8 infiltration. Again, they used a 30-pack year cutoff. I have to ask Vlad how they came up with a 30-pack year versus a 10-pack year, but all that to say, in the extremes, in the extreme smoker group, they saw differences with respect to CD8 infiltration using IHC. So looking at those data, the next step is, well, let's go deeper with this. How can we examine the tumor immune heterogeneity in HPV positive cancer beyond doing IHC for PD-L1 or CD8 infiltrate? So we comprised a cohort. We constructed a cohort here at WashU of about 50 HPV positive oropharynx cancers, where we did whole exome sequencing to understand the genomics of these tumors. We also did a nanostring immune oncology panel. This is an off-the-shelf nanostring panel of 770 genes that will give us some insight into the immune microenvironment in these tumors. We also did some IHC validation and did some validation in TCGA. And I should note that Paul Zolkind was a critical part at the beginning of this project and has kept contributing even from his time at Stanford. So I appreciate all his input and work into this. So these are our data from the whole exome portion of our study. We again asked the question that we asked in the Chance Carolina cohort that we had, whether smoking had an impact on mutational status. It did not. We did not find genomic differences between smokers and non-smokers. PIK3CA, FGFR3, zinc finger 750 were again the most common mutations that we saw. We also saw TRAP3 mutations, and we report B2M mutations that were previously reported in TCGA, but not statistically mutated before this cohort. We also see B2M mutations that are important in antigen presentation, incidentally, in the Chance cohort as well. We also used our data to understand the mutational signatures again, because I think this gets to the heart of carcinogenesis in these cancers. We again found that aging and APOBEC signatures 1, 2, and 13 were the most commonly the dominant signatures in these tumors. And again, we found very little evidence of tobacco-associated mutagenesis in these patients, even among the smokers. So more evidence that inherently these tumors at early stages of carcinogenesis are not being driven by tobacco or by tobacco carcinogens. They're being driven by the virus. Now, whether or not the smoking has an impact on tumor infiltration is the next question that we asked. But it's important to note that even in two cohorts now, we've shown no genomic differences or no evidence of early carcinogenesis driven by tobacco and HPV-positive cancer. So the next question was, well, what can we do with our RNA sequencing or our nanostring data to help understand the landscape in terms of tumor immune infiltrates in these tumors? And we did some non-supervised clustering that led to some interesting findings with respect to T-cell activation among non-smokers and an association with better outcomes. But then we came across the TGap score, which is a pan-genomic marker designed to give insight into PD-L1 checkpoint response. And this is a marker that's been developed now across multiple cancer types. It's agnostic to tumor type. It's an 18-gene T-cell inflamed score that allows you to predict response to immunotherapy, namely Pembro in the case. This is proprietary to Merck. And what they are able to do is able to combine the gene expression of these 18 genes that are critical to Pembro response with mutational burden. And although each of those variables give some prediction of response, the combination of mutational burden with this 18-gene TGap classifier provided prognostication, but more importantly, provided response to Pembro. So when we looked at our cohort, we looked at the TGap score in this cohort of 50 HPV-positive cancers. And we did in fact find that the TGap score was predictive of survival. So not only is TGap predictive of response to Pembro, but in a cohort of patients treated surgically with adjuvant radiation and chemotherapy, we found that the TGap score was prognostic in a way that was agnostic to treatment type. So even standard of care therapy, we found that T-cell infiltration had a significant impact. We also found that TGap was affected by tobacco use. So the TGap score was significantly higher or more immune infiltrate, more T-cell activation in never smokers compared to former smokers, compared to current smokers, as you can see here in this bottom graph, in a dose-dependent fashion. So after all this work, we started seeing a signal showing us that even though there's no genomic differences by smoking, we're finally getting into some understanding of how the tumor immune microenvironment is affected by tobacco and how that potentially can lead to different outcomes. So we also were able to understand, again, based on tumor mutational burden and the TGap profile, able to classify our patients across these 4 quadrants. And we found again that the never smokers were the high TGap, high mutational burden patients who also happen to have the best outcomes. You can see on the right, our graph here from our paper showing that the never smokers fit primarily into that upper right quadrant of high TGap, high mutational burden. We then were able to validate this across platforms and across cohorts. So our platform was a nanostring platform here at WashU. We looked at the RNA-seq data from TCGA, and we found very similar findings where the TGap was predictive of outcomes, which was not surprising, but also associated with tobacco. And finally, we were able to look at TGap with respect to PD-L1 expression. And as predicted, we saw a direct correlation between not only PD-L1 expression and TGap, but also CD8 infiltrates in TGap, giving us more understanding of how important smoking is as a modulator of the tumor microenvironment for HPV-positive cancer. And as I mentioned, TGap was associated not only with overall survival, but with disease-specific survival in HPV-positive cancer. So although TGap has been looked at in multiple different cancer types, it hasn't really been looked at prior to this study, specifically in HPV-positive cancer. And TGap is an attractive classifier for tumor immune infiltrate because it's been validated in multiple cohorts. And we think it has the potential to serve as a future biomarker or inclusion criteria for treatment de-intensification that's probably superior to smoking alone. I should note that there were some smokers that had high TGap score, and there were some never smokers that had low TGap scores. So this marker probably has more potential than what we're currently using to define who deserves treatment de-intensification, namely smoking, because it gives us insight not just into a patient-derived variable, but how that variable actually impacts biology of these cancers. So the question is, how do we incorporate this into the future trial? So this is our trial that we just finished accruing for in HPV-positive cancer here at WashU. We treat HPV-positive cancer here primarily with transoral surgery, about 90 cases a year. We have incorporated this MINT protocol into our practice, and we just closed this trial where patients were treated with 42 gray alone in the lowest risk patients, 42 gray plus cis in the intermediate risk patients, and then 60 gray all in the adjuvant setting, of course, in the highest risk patients. So as we think into MINT2, we wonder whether or not we should incorporate something like a TGap score or PD-L1 expression into this, whether we incorporate immunotherapy based on such score. We're currently thinking along those lines for our next de-intensification study. And I should note, this is not this trial that we just finished accruing. We have not had a single relapse event in arms 1 or arms 2, and this was agnostic to smoking status. So in conclusion, I think it's clear that smoking has a profound impact on the entire arc of HPV-positive cancer, from carcinogenesis or from infection of HPV all the way through carcinogenesis and all the way through treatment and outcomes. And although we haven't found significant mutational differences between HPV-positive cancers and smokers and non-smokers, we did find these interesting viral subtypes that I think will emerge as important biologically when we start defining prognosis. And we're not the only ones, of course, to show this. Maura Gillison's group has shown this in cell lines, but the unique aspect of our DNA technology of the sequencing cohort gave us a strength to look at these viral structural subtypes that I think will become more important. And the TGap score represents one of multiple prognostic biomarkers of the tumor immune microenvironment that we should consider incorporating into future trials. Not only trials where immunotherapy is involved, but trials where standard of care is involved and de-intensification is considered. So where would we go from here? I'm working with colleagues in Houston we put together a grant to look at these aspects and validate some of our findings, but also look at this using a mouse model. Also look at markers of the systemic immunity by smoking status with Andy Sikora at MD Anderson. And we're incorporating the TGap score into corollary studies on MINT1 on our trial here, and further expanding our cohort. So I want to acknowledge all of my co-investigators, as well as the folks who have participated in my lab here at WashU, including, as I mentioned, Paul Zolkind, as well as funding from the NCI, American Head and Neck Society, and others, and all the collaborators that you see listed here, who obviously were a critical part of all the work that was presented here today. So thank you all very much for your attention, and I'd be happy to take any questions.
Thank you so much, Jose. Great lecture.
Thank you.
Let's see if there's any questions coming through the chat.
So I'm gonna try to preempt Dr. Sun Wu before he asks a really intelligent question. So my question is on the, you talked a lot about the immune score that Merck had developed and looking at that. I'm kind of curious if that tells us anything biologically, or is that just a sort of empiric data, and what that might reveal for head and neck cancer specifically. And it sounds like it's a pretty widely used, marker for multiple tumor types, but.
Right, it's a good question. The score primarily gives us information about T cell activation and T cell infiltration. And I think that's represented also in the PD-L1 expression, IHC. So it correlates very clearly with PD-L1 expression in these tumors. But it also incorporates tumor mutational burden, which gives us some idea of the heterogeneity of these cancers. But to your point, I think it requires a lot more validation in specific subtypes. So there's publications now that have looked at this signature in esophageal cancer, in other cancers, to hone in on more of the heterogeneity that these represent in specific tumor types, because I think it's valuable because it's been validated and as a response to immunotherapy, but I think there's a lot more work to be done in each of these subtypes. And part of what we showed today is looking at this specifically in HPV positive cancer.
Jose, this is John. It's a great talk. I really enjoyed it. I found your analysis of the integrated forms of HPV versus the episomal form to be really, really interesting. I was just wondering if you compared primary versus lymph node in a particular patient, whether or not there are differences of the episomal versus integrated form, or if it's consistent in the primary versus the lymph node.
So that's a great question. We have not done that study yet. And I think one of the valuable parts of a study like that would be that when we look at these tumors, not every cell in any given tumor is all integrated or all episomal. So I think these are oftentimes mixed. And it would be interesting to know whether or not a lymph node metastasis has a different pattern of structure than the primary tumor. But it's an interesting question that we haven't looked at yet. But thanks for the suggestion.
The other question I had was something that's really interesting to me is about the HPV negative non-smoking cohort. And I know this was data that you had pulled from, I think, some collaborators, but correct me if I'm wrong, but did you say that the non-smoking cohort also had a significant number of mutations that were, and that they weren't different from the smoking cohort? Is that correct?
So in oral cavity cancer, the mutations were similar. They did not see the mutational signatures and actual mutations between oral cavity smokers and non-smokers, HPV negative, were quite similar, but the differences lie in the immune classifiers, similar to what we're seeing in HPV positive cancer. Got it.
And sorry to not to hog the microphone here, but one other thing that I think would be really interesting in your cohort to look at is whether or not there are differences in antigen presentation between the integrated forms of HPV versus the episomal. I think the integrated form it may yield a different pattern of neoantigens or peptide MHC presentation compared to the episomal form.
Right, and that's aim 3 of our new grant. So I'm glad you brought it up. And it's exactly what we're doing now with this exact cohort is we're looking at the transcripts and looking at neoantigens as well to see what these structural subtypes actually mean with respect to neoantigens and also with on the RNA level. So I think that has important implications as well, because there's probably an immune response that's different between these different subtypes of HPV. Our first step is to, again, validate these structural subtypes with long read sequencing and other methods, but also to look at neoantigens and look at transcripts. So that's another very good point.
That's a great idea. Well, if we can help you, what we're doing is we're profiling the peptide MHC immunopeptidome by mass spec. So if that's of help to you, we'd be happy to help.
We should talk. I think that would be great. Thanks, John. Chris, I see you moving, but you're muted.
So the immunopeptidome, isn't that the name of a band that we were going to start, the immunopeptidome? I thought if you do start a band, John, that was what I would use for the band name, for sure.
Great name.
That's a great idea. I'm going to start making T-shirts now.
Hey, Jose, can you hear me okay?
Again, hi, Chris.
Hey, wonderful talk. And I also, want a T-shirt as well for the band. I think that you're really right to start your research out by questioning this sort of 20-pack year, 10-pack year, and then Vlad is, advocating for 30-pack years. It's such a great illustration of the story of precision medicine, where we estimate using a crude clinical measure of a disease, but you're drilling down and finding that biomarker. So this is exactly what Creighton Christensen had in mind when he coined that phrase, precision medicine. So, wow, my hat is off. And, I remember you as a junior resident over at Baylor. And so it's really amazing. Congratulations on your journey. I do have one quick question for you. How robust would this TGap be across institutions to help us, if it's a great biomarker? Is it going to be something that is as readily standardized and useful as a CPS score or traditional HPV-ish? How robust do you think it would be as a biomarker, Jose?
So thanks, Chris, for your comments and for the question. I think currently it's not widely available. What they, I think, smartly did was they built the platform using NanoString, and there are examples of NanoString, CLIA-approved tests that one could order now. So I think in the future that could be something that could be used. And TGap, I should just mention, is one example, right? That happens to have a Nature Medicine paper and works across multiple cancer types. But it may be that a better signature is out there that shows us more or gives us more refined understanding of the tumor microenvironment. So I think some version of this, to your point, is going to trump something like smoking, or especially when we're talking about pack year history, right? It's amazing that, we can do these really interesting analyses about the genomics of tumors, and we're basing the entire premise of a study on whether the patient remembered he smoked 10 or 20 pack years, right? So I think we just need to do better, and this is one potential avenue by which we can do so.
But it was interesting, I appreciate Chris's question, because a little bit I had the same question. When I looked at the bars, they just looked grossly overlapping. They were so, I know they achieved statistical significance by averaging, but you look at it, they're so overlapping that it'd be hard to say for any one patient what the results are. So I think that's what Chris is getting at. And I, maybe if we combine 6 of those kind of measures, so as blunt as they are, sometimes the smoking, is an interesting way to look at it. But I guess that was what I would want to know about what it's telling us about the biology. But as a marker, it just seems like there's too much overlap for an individual patient.
Right. And, this was looked at over 5,000 patients in multiple, 4 different trials in the recurrent metastatic setting, right? So this is a beginning of understanding the prognostication as opposed to just understanding response to immunotherapy. So there's a lot more to learn about this signature, but I'll stress that the concept of the tumor immune environment being impacted by smoking, not just because of TGap, but because of our other work, is relevant, but has to be, more clearly defined. There's no doubt about that.
And just to flesh out some of the historical aspects of this, it's really important, especially for the residents listening, is that, the Ang paper was a recursive partitioning analysis, a post hoc, unplanned secondary analysis of only 270 patients in that trial. And so 20 pack years doesn't really work because it wasn't a planned analysis. And yet we sort of latched onto it. And then in the next trial from the RTAG, they went with 10 pack years, and Vlad's moving it to 30. All of these things are subjective, which just emphasizes the point more that keep moving full steam ahead with your work so you can find a more robust biomarker. Jose.
Thanks, Chris.
Well, I think that's it as far as questions. And once again, Jose, great to see you. And thanks so much for coming virtually for Grand Rounds.
Thank you very much, Tulio. And thank you all very much for your attention. And I hope to see you all soon in person.